replication reflexes travel to the brainstem to produce abdominal, colonic, and rectal contractions. So those are electrical, nervous controls, feedback controls for the GI tract. There are also hormonal controls. So these are hormones that are released into the bloodstream, which have an influence on gastric motility, or sorry, not just gastric, GI motility. And these include gastrin, gastrin, which is secreted in the stomach in response to distension, the presence of proteins, and the release of gastrin-releasing peptide, which is released due to parasympathetic stimulations. This is stimulation by the autonomic nervous system. Gastrin stimulates gastric acid secretion, and also is involved in the growth of the gastric mucosa. CCK is spell it cholecystokinin. And this is secreted in the duodenum and the jejunum in response to the presence of fat and fatty acids. And this causes bile to be released into the small intestine to break down its fats and released from the gallbladder. It also inhibits stomach activity, so it gives more time for this bile to work to break down these fats. And so that might be something you've noticed if you eat a meal of which is fat-free. You tend to get hungrier quicker than if you eat a meal that's very high in fat. It takes longer for the stomach empty to occur. GIP is gastric inhibitory peptide, which is secreted in the upper small intestines, in the duodenum, the first part of the jejunum. Again, in response to fatty acids, amino acids, and carbohydrates, and this again inhibits stomach emptying and motility. Secretin secreted by the duodenum. So again, it's the first section of the small intestine in response to acidic gastric juice. So, stomach empties acidic fluids. As the stomach empties, contents are acidic, so that as the duodenum becomes more acidic, it secretes secretin, which promotes the pancreas to produce bicarbonate ions, which neutralise the acid. And lastly, it's motilin. Motilin, which is secreted by the duodenum during fasting. And this promotes what's called the motor, migrating myoelectric complex, MMC, during fasting, which has a sort of a housekeeping function. See, there's numerous methods by which the gut is aware of its has a role in increasing motility during fasting. So it's released when the stomach empty and the gut is empty. So for some of us, it never happens. For others, at night, most common. And this, I'll come back to it if we've got time. But there's a particular characteristic contraction pattern which moves through the entire gut, the MLC. So we have numerous ways that the gut coordinates the contraction and the movement of food through it. This shows, actually, I'll come back to the bearings. So these reflexes inhibit or enhance activity. But that activity is there all the time. So if we look at the stomach, these are electrical activities, so depolarization of muscle cells. And like the cardiac pacemaker, which we talked about last week, these are automatic cells. They produce these electrical activity, these slow waves, automatically. And what this part of the graph shows is the activity in the various portions. And we can see there's a wave of activity which moves down the stomach towards the pylorus. Now what these are changes in membrane potential. Which give this rhythm to the gastric and gastrointestinal contractions. These are called slow waves. And depending on where you are in the gut, they have different frequencies. So it's about 3 per second in the gut, about 12 in the duodenum, something like 8 or 9 in the ileum. And normally, this membrane potential is minus 50 to minus 60 millivolts. And in most of the gut, it doesn't produce any contraction of the muscle. It's just a movement in the membrane potential. But when it starts to get close to minus 40 millivolts, we start to see spike potentials. So we see these spikes, and it's these spikes that initiate contraction of the muscles. So the higher, if the whole of this drifts up, we're more likely to see spike bursts. So we're more likely to get contractions. And the higher this potential is, the greater the frequency of these spike bursts. So we have these slow waves, and what happens is, due to our feedback, our reflexes, they can have an effect on membrane potential and cause these slow waves to initiate contractions. So stretching of the mucosa, presence of neurotransmitters, such as acetylcholine, which is released by parasympathetic stimulation, presence of some hormones will cause depolarization, so the whole thing becomes less negative and more likely to see spike bursts or excitable. Conversely, adrenaline, noradrenaline, released by sympathetic stimulation, can cause hyperpolarization. So the whole lot drifts, more negative, spike bursts become less likely, so we're less excitable. essentially the movements come in two flavours, two purposes. Peristalsis and mixing movements. Peristalsis is propulsive. Mixing movements function is, as the name would suggest, to mix contents to ensure good contact with the mucosa. So peristalsis, usually the stimulus would be distension, so there's the presence of a bolus stimulating these mechanoreceptors, and these are communicating with principally the submucosal plexus, which is also communicating with the myenteric plexus, which is communicating with these muscle fibers. And what it does is inhibit the muscles downstream, so it's relaxing these muscles. So the membrane potential is becoming more negative, less excitable. And it's exciting the muscles upstream. So we get receptive relaxation, contraction of the lumen, and this moves the bolus along. Usually it's from oral to anal, but these movements can move in the opposite direction. They usually die out very quickly. It's probably thought to be due to some kind of polarization of the plexus. And these may, in the small intestine, move perhaps 5 to 10 centimetres. Usually, another peristaltic movement goes further along. Mixing movements are mediated in the same way, locally constrictive contractions. So there's a contraction every few centimetres, which lasts a few seconds, up to perhaps half a minute, and then these die out and new contractions appear in different places, so it's the chopping and mixing context. As I say, this encourages contact with the intestinal wall, with the mucosa, to allow absorption. Top end again. Swallowing or deglutition has an oral phase where you take a bolus of food, you push it to the back of our mouth, and into the pharyngeal phase. So the food, as it reaches the back of the mouth, stimulates the epithelium there, swallowing receptors, and these impulses pass to the brainstem, which then coordinates the automatic muscular contractions that follow. It inhibits the respiratory centre, so we stop breathing at any point in the respiratory cycle. Food moves down into the pharynx towards the esophagus. So we reach the esophageal phase. Now the upper esophageal sphincter has been closed. In the esophageal phase, the pharynx contracts, pushing the bolus further down. The upper esophageal sphincter relaxes, the epiglottis closes over the larynx to seal it, and bolus of food passes into the esophagus. Then there's, once in the esophagus, there's a primary peristalsis phase. This peristaltic wave that pushes the bolus towards the stomach, which typically takes 8 to 10 seconds, or if gravity is helping, 5 to 8 seconds. That's initiated by this reflex from the oral phase, pharyngeal phase, esophageal phase. If not all of the food is passed in that primary phase, then there's secondary peristalsis, which rather than being initiated by this swallowing reflex, is due to distension of the esophagus. So it'll continue to move the bolus, the remaining food, by peristalsis. And as that peristaltic wave reaches the cardiac sphincter, it causes relaxation of the cardiac sphincter, which is the lower esophageal sphincter joining the stomach. And the stomach itself relaxes to receive the bolus. Go back 1 or 2. What this is attempting to show is the pressures within the esophagus during the swallowing phase. So, 1 in the pharynx is the pressure, which is dotted solid line, which is this one. So, at rest at 0 is where we begin to swallow, and there's a peak pressure as the bolus passes through the pharynx, which then relaxes to 0. So this is the upper esophageal sphincter, which is a dotted line, this one, so it's in tonic contraction most of the time, so it's closed. As the bolus passes, it drops down to 0 and then recovers. So it's a coordinated relaxation to allow the bolus to pass. 3 is halfway down the esophagus, 4 is towards the bottom of the esophagus, and we can see there's usually at rest, there's an increase in pressure as the bolus is passed. So there's a wave of pressure passing down the esophagus. And 5 is the lower esophageal sphincter, which again is usually contracted. As this wave approaches, it relaxes and then recovers to close. Lower esophageal sphincter is normally contracted, it's normally closed. In addition, the last portion of the esophagus protrudes slightly into the stomach, so the pressure within the stomach also keeps the esophagus closed to prevent reflux. So it stops the contents of the stomach, usually from coming back into the esophagus, where the acid and digestive enzymes will damage the esophageal mucosa. And it also resists high abdominal pressure, so it prevents reflux when we cough, breathe heavily, walking and so on could cause reflux, apart from the fact that the esophagus protrudes slightly into the stomach. See the stomach, as I've said, the cardiac sphincter at the top is a layer of oblique muscle, longitudinal and circular muscle, so it's different from the rest of the gut in that respect. And its purpose is the storage and digestion of stomach contents to produce chyme, which is passed into the duodenum. There's what's called the vagovagal reflex, which is from the stomach to the brainstem and back, which helps to reduce the tone of the muscle in the stomach in response to stretch, so it relaxes as the stomach fills. contents. The limit is usually about 0.8 to 1.5 litres. And the stomach's been empty for some time, several hours at least. We start to see rhythmic contractions in the body of the stomach, which can sometimes fuse together and last for several minutes. These can lead to mild pain or hunger pangs. They're most prominent in the young and healthy who have a high degree of muscle tone. Usually start 12 to 24 hours after the last meal and peak in intensity after 3 to 4 days, apparently. So the stomach itself has these characteristic movements. So propulsion, which is essentially peristalsis that we've discussed. So the upper part of the stomach contracts and forces food contents forward against this closed pyloric sphincter. This is closed and then can pinch off the antrum, which is this muscular portion at the bottom of the stomach, and produce grinding movements which help to break up the contents, mix these with the acid and the secretions, and then there's a period of relaxation where these contents move back up the stomach before the process starts again. Stomach empties into the small intestine. The small intestine, as I said, consists of the duodenum and the ileum. I've got bored of drawing them, but all the way down are these circular ridges, plicae circulari, which are covered in villi, these small needle-like protrusions. What this does is increase the surface area available for absorption. So there are strong peristaltic contractions of the antrum, which is also called pyloric pump, generating high pressures. Normally, if the pylorium is not unusually contracted, that will eject several millilitres of chyme into the duodenum for each contraction. And it's normally open enough for water to pass easily, but prevents food particles from passing until they're fully mixed. The regulation of stomach emptying, it's feedback again. Distension of the stomach increases peristaltic activity, so increases emptying. The presence of food, particularly meat, will increase gastrin production, hormone which stimulates acid secretion, stomach activity, so again increases emptying. There's an enterogastric nervous reflex from the duodenum. There's a nervous reflex from the duodenum back to the stomach, whereby increased volume in the duodenum stimulates a reflex via the myenteric nervous system, so alveolus plexus, but also the sympathetic prevertebral ganglia, so close to the spinal cord and back. And these all inhibit the pyloric pumps. They slow down stomach emptying. So if the duodenum is full, it will reduce emptying. There's also hormonal feedback from the duodenum. So fats entering the duodenum, fats are foods, will trigger the release of hormones which inhibit the pyloric pump and increase sphincter tone. So the pylorus closes, the stomach becomes less active, emptying decreases. This allows more time for digestion of these fats and so on. these reflexes, these enterogastric reflexes, respond to distension of the duodenum, irritation of the duodenum, acidity of the chyme of the duodenum, and the presence of breakdown products of proteins and fats will all reduce stomach empty without more time.
What was the last one you said?
The presence of proteins and fats will breakdown products of them. So, if the duodenum is sensing that it needs more time, it's inhibiting the stomach. Below 3.5 to 4, the pH of 3.5 to 4, further emptying of the stomach is blocked completely until the acidity is neutralised by release of bicarbonate ions, mostly from the pancreas. So peristalsis and mixing movements occur all the way through the small intestine until they reach the ileum and the ileocecal valve. So there's still liquid, essentially, at the ileocecal valve. The movements in the small intestines, I'm going to have to cover it. In the small intestine, we see peristalsis, which we've discussed. So they move a few centimetres per second in the proximal intestines, which is the duodenum close to it, and half a centimetre a second in the terminal intestines, so down by the ileum, the ileocecal valve. The average net movement is only about 1 centimetre per minute. In addition to peristalsis, we see pendular movements, so these are mixing movements where there's reverse peristalsis for a short distance, and also segmentation. So again, this is chopping and mixing the contents. Generally, activity will increase after a meal. This is due to, again, gastroenteric reflexes. So distension of the stomach increases peristalsis in the small intestine. See this is in a dog, so the numbers are slightly different in humans, but you can see, as in humans, a higher rate of contraction, so more slow waves per second. In the duodenum, it drops in rate through to the ileum. I mentioned the fasting motility, this migrating myoelectric complex, which is on the bottom. What this shows is various portions of the small intestine, the percent of slow waves that actually lead to contractions. So in this portion in the ileum, we still have slow waves, but they don't reach that threshold potential, so they're not causing contractions. And this is after fasting for several hours. There are periods which last quite a long time, perhaps an hour, where there's contractions produced by these slow waves, without any contents. And these move in sequence along the duodenum, jejunum, and the ileum, so they're moving through the system. It's thought to be mediated by motilin and have a function of housekeeping, so it flushes out any undigested stomach contents or matter that can't be digested, broken down. And then when we eat, when we have a meal, we return to normal normal motility pattern. So the chyme is spread out along the mucosa around good contact. We've blocked at the ileum for several hours until we eat. At which point there's the gastroileal reflex, so it's from the stomach to the ileum, increases peristalsis in the ileum, which forces chyme through the ileocecal valve into the cecum at the beginning of the colon. So that valve is normally mildly constricted to prolong the stay of chyme in the ileum for absorption. Typically about 1.5 to 2 litres of chyme empties into the cecum per day. If the cecum is irritated or distended, so distended because it has it's full, or irritated, so through an inflamed appendix, it will cause contraction of the sphincter and inhibition of the ileum peristalsis. So this stops emptying into the rectum, into the wrong part of the diagram, into the cecum. Now the colon essentially has similar structure to the small intestine in terms of the muscle layers, the plexus and so on, but functionally looks rather different because its job is different. What the colon does is absorb water and electrolytes from the chyme and stores fecal matter until we're ready to expel it. So it has more sluggish movements, but like the small intestine, there's also propulsive movements and mixing movements. Now the mixing movements called horstrations. So there's a horstra, these bulges in the wall. And what happens is a section of the circular muscle will contract almost to occlusion, about 2.5 centimetres long section of circular muscle. And other sections of circular muscle further along will also contract. And there are strips of longitudinal muscle called the tenea coli. I'll show them right here. So there's 3 strips of longitudinal muscle which also contract. And these have the effect of, these contract the unstimulated portions between these longitudinal muscles, bulge out and form these horstra, which has the effect of moulding the feces and ensuring good contact, so chopping. Peak intensity of these horstrations is reached in about half a minute and disappears over the next minute or so, and then a few minutes later new horstrations will appear nearby, so there's a mixing effect and also a small propulsive effect. So as we go through the colon, we start from fluid, semi-fluid, mush, semi-mush, and solid until we reach the rectum where we have solid feces, and about 80 to 200 millilitres of feces is expelled each day from a starting point of 1.5 litres to 2 litres of chyme. So most of the propulsion is accounted for by these horstrations, but there are also propulsive movements called mass movements, constrictive ring which appears in the circular muscle in response to a distension or irritation, very often in the transverse colon, portion of the top. And then 20 centimetres or so of colon distal to the ring, so downstream of the ring, will lose their haustra and will contract as a unit as propels the faeces forward. And these again peak in intensity for about 30 seconds, releasing after 2 or 3 minutes, and then repeat further along. So these series of movements will persist for 10 minutes, half an hour, and when sufficient faeces have been moved into the rectum, there's a desire for defecation felt. These propulsive movements are initiated by gastrocolic and duodenocolic reflexes. These are signals from the stomach and the duodenum which stimulate due to distension the formation of the initiation of these propulsive movements. And these are transmitted by the autonomic nervous system. Persistent mass movements sometimes occur if there's irritation, long-term irritation of the colon, ulcerative colitis, which causes more rapid than normal movement of kind through the large intestine. So when sufficient solid has been propelled into the rectum distends and initiates a reflex contraction. And in addition, initiates relaxation of the anal sphincter. So we have the rectum and anus, and there are 2 sphincters, the external and the internal. The internal anal sphincter is smooth muscle, which is under automatic control, controlled by the nervous system. The external anal sphincter is under voluntary control. It's striated muscle. Probably more accurate to say it's under subconscious control. It's normally contracted unless it's voluntarily relaxed. So there are reflexes controlling defecation, distension of the rectum, initiates peristalsis in the descending colon, in the sigmoid of the rectum, and relaxation of the internal sphincter. And this is mediated by the myenteric plexus, so it's the enteric nervous system. There's also parasympathetic stimulation, so via the spinal cord, so this distension initiates parasympathetic stimulation, which intensifies the peristalsis and internal relaxation of the internal sphincter, so reinforces the intrinsic reflex. So if once these reflexes start, we still have voluntary control, but if it's convenient, we can relax the external anal sphincter and defecation will occur. There's also communication with the spinal cord from this reflex, which initiates other effects, deep breath, closed glottis, contraction of the abdomen, which enhances the peristalsis.